Maria Kristanti Ahindi, and today I will present about modeling meiosis. A little bit about meiosis. Gametes in humans and plants are produced through meiosis. Two primary purposes for meiosis are the chromosomes number is reduced from diploid, it's 2n equal to 46 in humans, to haploid, and is equal 23 in humans, and genetic diversity is promoted. And two main sources of genetic diversity are crossing over during prophase 1 is homologous or non-sister chromatid swap DNA, and the second one is independent assortment of the homologous pairs. They do not always line up the same way. So for this simulation, I'm using blue, red, and white beads, fuzzy sticks, scissors, papers, pen or marker, ruler, and table. So using beads and sticks, I'm making chromosomes and centromeres. So the centromeres are presented by these white beads, and the different colors represent homologous chromosomes. So this is my original cell. It also called the parent cell. As you see here, there are 4 chromosomes. We say the diploid number is 4. So as you see this, there are 2 short and 2 longs. These 2 longs are homologous to each other, and these 2 short chromosomes also are homologous to each other. That means the red one came from the mother, the blue one came from the father. They are the same size, the same shape, and they have kind of similar genes, but they are not exactly the same. So as I mentioned, this the short one, the short chromosomes are homologous to each other, and again, the long chromosomes are homologous to each other. So this all is in G1. They are doing regular cell jobs. So what comes after G1 is S phase. What happened on this phase? Everything doubles. As you can see here, everything's double. All the chromosomes replicate. Then after S phase, what comes is G2. In this phase, the cell is growing, getting prepared for cell division. After G2 comes meiosis. What makes meiosis different is that there are 2 sets of division. There is meiosis 1 and meiosis 2. And we need to be specific when we're talking about whenever we refer to any of the stages. So let's start with the meiosis 1. So on meiosis 1, the first step is called prophase 1. So on this phase, the nucleus goes away. In prophase 1, besides the nucleus goes away, there is something else happened in this phase that didn't happen in mitosis. So what happened is here is something important, which is called crossing over. What that means that the chromatids from homologous chromosome will actually cross over. So it's going to be break off a piece of themselves and exchange portion. And you can notice here 2 different color crossover. So remember, these 2 are identical to each other, and they cross over. So you can see here on the picture the different color, both to the long chromosome and the short chromosome there. So these 2 are identical to each other, as I mentioned before. It has to be the chromatids that are not identical, so they are now in different color. So remember, because our parents were not related, these two chromatids, right, are noted the red and the blue are not identical to each other because they came from different parents. So, okay, it's a crossing over here. It can be just a little, or it also can be the whole branch crossover of our chromosome. So it also can happen more than one. So again, on this phase, prophase 1, there is crossing over. So that's the end of prophase 1. The next step is what is called metaphase 1. So on this phase, we have fibers come out and also they line up along in the middle. So they line up in a single line as you see on this picture, double up, and this set is random. What does mean? They line up in different color. It can be in different color, but still in the single line in the middle. So this line up process, as mentioned before, is random, and this is what is called independent assortment. So this big thing happened here during metaphases 1, again is called independent assortment. What comes next is anaphase 1. So on this phase, it separates process. So what separates now is homologous chromosomes separates from each other. So again, this is the process of anaphase 1. After anaphase 1 comes up telophase 1. On this phase, new nuclei is formed. So this becomes a nucleus and this is also another nucleus. So this is the telophase 1. The next step is cytokinesis 1. So what does it mean on this phase? It's become a new cell. So after meiosis 1, we will have two cells now. Each cell only has two chromosomes. So we went from having four chromosomes per cell, then to having half that, which is two chromosomes in each cell at the end of meiosis 1. So each cell is now, we say this is haploids because it has half the number of the chromosome than when it started. So again, each cell is now considered haploid. After this cytokinesis 1, we will start the meiosis 2. As meiosis 1, in this meiosis 2, we will also have the step of prophase, metaphase, anaphase, telophase, and cytokinesis. Let's start our meiosis 2 process. So in meiosis 2, it will be started again from prophase 2. So what happened that each of the two cells now goes through meiosis 2 at the same time. So on this prophase 2, what happened is that nucleus disappear again and makes fibers again, but this time is no more crossing over. There is no more crossing over anything because the homologous ones have separated, so we cannot cross over anymore. So this is the prophase 2. Next step is metaphase 2. Again, on this phase, they line up along the middle. Now with the fibers make them line up. So again, it's the straight line again here. So this is the metaphase 2. Comes after metaphase 2 is anaphase 2. So on this phase, as you can see on this picture, the chromatids that separate from each other, just like in mitosis. So again, on this phase, they separate. So after that is telophase 2. So the new nuclei start forming again here and become a nucleus. So now there are 4 nucleus on this telophase 2. Then finally, we come to cytokinesis 2 phase. So on this phase, it's become a new cell. So now we have 4 new cells in this cytokinesis 2. By the way, this is what is called daughter cells because they are the result of a division from the original cell, which called a parent cell. To conclude this simulation, that mitosis making exact copies and meiosis making daughter cells that look completely different, and each daughter cell only has half the number of chromosomes as the original cell, and it's from diploid to haploid. Every single sperm and egg is genetically unique. And also, during meiosis, when it introduces the genetic variation. Thank you for listening my presentation. I hope you enjoy.